I, the chairman of the foundation, Dr. Mahesh Arvin, welcoming all the participants for this talk. The speaker of the day is Mr. Varadaraj, who has come all the way from US to India to his native place. And our foundation has taken an opportunity to make use of him for this talk. So before he starts his talk for the day, I introduce him. It's indeed my pleasure to introduce today's speaker, Mr. Varadaraj. Mr. Varadaraj hails from Kanakapura district, Karnataka, India. He had his graduation from Bangalore University, then post-graduation from Nrupatunga University. After completing his MSc in organic chemistry, he worked as an assistant professor of chemistry. Then he left India for his research studies. He joined Indianapolis University last year, that's in the year 2024, as a research scholar. His research studies is on new ambient ionization techniques under the guidance of Dr. Nicholas E. Mankey. Today's talk is relevant to his research studies. On behalf of Prajna Foundation, on my own behalf, and on behalf of all the participants whoever have joined now, I welcome him for the talk. Welcome,
Thank you, sir.
Mr. Varadaraj.
Thank you for
Over
the
to
great
Varadaraj for the talk.
welcome. Thank you. I'm just now sharing my screen. Is it my screen is visible for you guys?
Yeah, now.
Yeah, fine. So, good evening, everybody. So, today's talk is about mass spectrometry research in ambient ionization. So, as sir
So
mentioned,
please show your face once. Let the
okay, fine.
audience be aware of you.
I think my photo is there in second slide. It's fine. Sorry. So, mass spectrometry research in ambient ionization. So, as sir mentioned, I'm the fellow who done masters in organic chemistry, then moved to analytical chemistry, being a PhD student. We works on mass spec, especially in the method development in ambient ionization. I think in India, ambient ionization is a word which feels very different because nobody knows how it works. But generally, once we come across the mass spec word, we think it's just LCMS mass spec, like liquid chromatography connected to mass spec, just for mass analysis. So, in this talk, I'm going to talk about introduction to mass spectrometry, then schematic representation of mass spectrometry, how it's look like, what are the main parts of it, then ambient ionization and its types, then three important ambient ionization we come across in our lab: disabsorption electrospray ionization DESI, direct analysis in real time DOT, and paper spray ionization. And the one of the main important thing I'm going to talk about is Faraday ion concentration polarization FICP, then how we connect FICP in the real time of science, and conclusion about this talk. So first, mass spectrometry. Mass spectrometry is a device which analyze any molecules based on mass to charge ratio. Mass means a molecular weight to charge, MOC. The molecule may be at any concentration. It may be ppm level to parts per trillion level. It may be in the any form. We can put it to liquid form or solid form. We can ionize them to the gaseous phase. Those gaseous phase ions getting into the mass spec and analyzed by MORZ, mass charge ratio, then we can qualitative, quantitative study of them. So compound like drugs, then warfare agents like TNT, then pollutants, in pollutants microplastic, PFAS, as any drugs. Otherwise, you can take pesticides, biological samples like proteins, lipids, metaboloids in a tissue. Otherwise, in a cell, anything can be analyzed in both quantitative and qualitative ways. So there are three main important factors in mass spec. One is ionizing a molecule. Ionization, being a chemist, everyone knows putting a charge to the molecule is called ionization. Creating a charge on a molecule in the gaseous phase, because we are ionizing, we are analyzing a molecule in a mass spec in gaseous phase. That's why creating a charge, both either negative or positive charge on a molecule is really important. So then mass analysis. We are analyzing those gaseous phase ions based on their mass, MORZ, then reduction of those respective MORZs. So now this is a schematic representation of how mass spec look like. This simple LTQ, linear ion trap. So in the right hand side, this is a first ionization. There are different types of ionization we come across in our day-to-day life. This, the one who doing mass spec really knows about these different types of ionization. The first one, electrospray ionization, is a gold standard method. Everywhere in the industry nowadays, in any pharma industry, they use electrospray ionization. It's a soft ionization. You can get a very good molecular ion peak. I will tell you what do you mean by molecular ion peak in further slides. Then nano electrospray ionization, which is less matrix effect, less noise effect. Then EI, electroionization, you can come in GCMS. Those who know GCMS, they know electron ionization. Then DISI, disabsorption electrospray ionization, ambient ionization. So ionization means you're putting a charge to your analyte molecules. Once those analyte gaseous phase ions getting into the mass spec, these ion optics are just accelerated by the different electric fields. They are just helps to guide the ions into the mass analysis. Now this entire system run in the vacuum system. Actually, here the mass analysis are the really important things in a mass spec. So there are different types of mass analysis depending upon the resolution we want. The mass analysis like quadrupole, ion trap, time of flight, linear ion trap, cyclotron. Here cyclotron resonance FTICR is one of the highest resolution based mass spec, and also time of flight is highly used for the protein based synthesis, and quadrupole and QX arbitrap is used for the proteins. Then once you ionize in the mass analyzer, the mass analysis is done, then they are getting into the detectors based on the different methods like microchannel plate, otherwise direct anode electron multiplier, just based on the how electrons strike into those plates, depending on the electric field, you're gonna get a mass spectrum. The mass spectrum is just a representation of M or Z in the x-axis and their abundance of respective ions in the y-axis. So now, what are the different application of mass spectrometry? Generally, in the medical field, mass spectrometry is like doing really good things. A reduction of metabolic disorders in a tiny blood spot. Generally, once a newborn baby is born, they will collect a small blood spot in a paper, then they will ionize the metabolites and they check for disorders and cancer diagnostic. If in case you look at the research in entire US, the cancer research is one of them. Generally, the image, any tissue imaging, cancer research by peptides, cancer research by proteins is one of them. Then therapeutic drug monitoring, how drug flows to the body. Then clinical toxicology, that's we generally works, reduction of drug abuse in both blood and urine, in like opiates, cocaines, methamphetamine, fentanyls, so many of them. Then identify poison and toxins in the human body by again urine and blood. So this how the LCMS look like generally, the Q exacto mass spec with X-ray and LCMS. Then and also the highly used in pharmaceutical industry for qualitative and quantitative analysis in analysis of drug structure by using internal standard and also analysis of drug metabolism. In US, in any company, actually they morely focused on the rules and regulation putting by the government called EHS rules to take any compound to market. They're gonna play with so many different rules, especially pesticides, herbicides, and industrial chemicals. To play with those things, mass spec really helps in so many different things, like how they works in soil, how they works in water. We can analyze the structure, what are the effect of them in the future, like that. And also air pollutants and microplastics, we can analyze in sub ppd concentrations. And also in usually in food industry to detect any contaminants and as well as any food adulteration, we can check through this. There are so many things like lipid detection, there are so many adulteration detection papers you can available in the ACS websites. So then proteomic study, we also do some proteomic study in our lab in ambient ionization to check the peptides and proteins and end-linked glycosylation sites to again study on the cancer research. And also forensic studies, analyze the gun residue powder and explosives. You're looking at the image. This is portable mass spectrometer from our lab. There's a paper spray portable mass spectrometer. You can carry to any destination you want, just like 15 to 20 kgs. You can ionize a molecule in a short period of time and you can get the result in this one. So, and also petrochemical industry research to just to check on the crude oil lipid sections. Then also space explorations means to check the soil and also other organic molecule present in it. And also in the biotechnology field, we use for so many things to just check the post-translational modification of DNA and otherwise RNA things. These are the basic applications of mass spec in a different field of science. You guys found any doubt? Happy to stop me by and ask a question. I'm happy to answer for that one. Thank you. Next one is the important thing I'm going to talk about is ambient ionization. What do you mean by ambient ionization is simple. I already mentioned everything go in a vacuum system. Ambient ionization is ionization taking place in atmospheric pressure means open air. No vacuum system is required. So there is no vacuum. We can directly ionize a molecule on the paper, otherwise on the tissue itself. So what's the main advantages of this ambient ionization means? A minimal sample prep. You no need to prepare more samples compared to other methods like LCMS or capillary electrophoresis mass spec, otherwise different mass spec methods. And also this real-time fast mode analysis, just 5 to 10 minutes. And cost effective because you're not spending more money on any sample preparation otherwise, and also the manpower. So these are the three different ambient ionization we come across in our day-to-day life. One is DESI, disabsorption electrospray ionization, DART, direct analysis in real time, and pepper spray. Here the images, how the DESI look like. This is the like the plate sample we are ionizing into the mass spec. This is a PSMS, pepper spray. This is DART. This is a paper. This paper consists of a analyte. We are placing analyte to the earth. This is jet method. I will explain in the future slides. So first one is DESI, disabsorption electrospray ionization. DESI is invented by Grams Cook. My PI is PI in Purdue in 2004. The representation of DESIs look like this. There was a jet section which is connected to nitrogen gas. Sir, some disturbance is going on. Someone need to mute actually.
So please, from the audience side, mute your audio, please. From the audience side, there is disturbance for recording also, as well for the presentation. Mute the audio, please. Sir, you can proceed.
Okay, so thank you. So here the way of how the DESI look like. Here the nitrogen gas or helium gas is coming through gas jet. So with the ESI solvent, then it is taking out to the surface of the sample, then getting into the mass spec. So again, it is a rapid mode of analysis. Actually, it's the first ambient ionization from Grams Cook lab in 2004. Then soft and surface-based ionization, you're directly ionizing the molecule from surface. Then there is no much sample preparation. Take your analyte, otherwise any tissue, Just put a electrospray solvent through jet with electric field in, then you can ionize it. And there is, you can do chemical imaging in DESI, actually one of the best ambient ionization for chemical imaging. Then generally used for chemical analyzing chemical warfare agent from the soil or water, then pharmaceutical compound analysis, and especially biological tissue imaging. How the mechanism works in DESI imaging. So here there is an electrospray solvent in the syringe. In the pump is pumping to syringe. That electrospray solvent generally a methanol water, otherwise acetonitrile water with some amount of formic acid or acetic acid, which helps for protonation. The protonation is main function because we want to create a charge droplets. These electrospray charge droplets are pushed into the surface by the nebulizer gas, that is nitrogen or helium, and also we are providing a positive volts or negative volts depending upon the which ionization you're doing. Once this charge analyte molecule strike onto the surface is contain our main sample. Once these charged primary droplets are strike onto the surface, then automatically those charged droplets will create secondary droplets. So these secondary droplets contain our analyte ions, then they getting into the mass spec, and then again MORC analysis, and we can detect what is there. This is how the simple mechanism of DESI look like. The electrospray solvent with help of nebulizer gas nitrogen or helium, or with high voltage electricity, they produce primary charge droplets. These primary charge droplets strike the analyte surface, means sample surface, creates a secondary charge droplets which contain our analytes, what analyte you're looking at. Then getting into the mass spec, then you just MORC analysis. Then DART, a direct analysis in real time. This is actually a rapid method compared to DESI. I loves to do DART in our lab. We have just now, like 6 months ago, we purchased this DART ionization source, which like cost around 40 to 50K dollars in US. So this image, how it's look like when connected to mass spec. This invented by Cody and Lauren in same Purdue University. Actually, this also second ambient ionization. This create metastable ions. Metastable ions means highly charged ions which strike onto the sample surface, then ionize them. Generally, here also helium or nitrogen gas is used to create those metastable ions. Actually, this really super fast. In some companies, they will do DART analysis, ambient ionization using robotic techniques. Then also this used for forensic studies, food safety and pharmaceutical industry and material analysis. So how the mechanism work like? Here, this is a simple representation of schematic representation of how that jet look like, the DART jet. So you are putting helium gas and nitrogen gas in the back, and the glow discharge which contain electrodes which connected to electricity. Those things will create in the heat system, they will create a metastable ions. Metastable ions are like, they are in between the ground in upper atomic state. They are not highly stable, they're highly energetic. They want to strike and lose the energy. Those ions, once they strike into the atmospheric components like water and nitrogen, because I already mentioned ambient ionization means we are ionizing the molecule in the atmospheric pressure. So once they strike into the any atmospheric component like water or nitrogen, they're gonna produce a protonated clusters. Once they just strike onto the our sample, depending upon the which ionization mode we are putting positive or negative, they're gonna ionize, then getting into the mass spec, again a MORC analysis. That is really fast analysis compared to DESI, but you don't get that like mode of very good result in terms of noise. So by looking at the low concentration, dark not work that much. So next one is paper spray ionization. Our lab do most of this because we are the group of paper spray ionization, because this one is invented by Dr. Maneki and Purdue under Graham's Cook guidance in 2009. Paper spray ionization is simple. Just take a piece of paper, putting into the cartridge, and also connected to the high voltage cable as I show here, which is depending upon the whether positive voltage or negative voltage you are putting in. Your analyte you're spotting like this, then you put a spray solvent onto it. What do you mean by spray solvent? Means I already mentioned ASI solvent. and 90 is to 10 methanol water with formic acid, otherwise 90 is to 10 acetonitrile water with formic acid. That's that spray solvent helps to flow your analyte to the paper tape. Once you connect it to the mass spec, that's going to produce a charged droplets. Then those things are getting into the mass spec. So once they're getting into the mass spec, we can just ionize. We use different types of papers, like 3180 cellulose paper means just cellulose filter papers, then glass fiber substrate, then PTFE substrate, then other different types of substrate. We generally use these three mainly. These PSMS is really fast mode of analysis, less cost effective compared to last two because everything is human made. Nothing is like you need high cost of purchasing also here. The paper you can get chromatographic paper easily. The cartridge is printed by 3D print cartridge. Everything is just a less cost effective method. So advantage same, minimal amount of sample preparation. Yes, you just to prepare you can just spot a whole blood, otherwise urine here, then you can ionize those drugs, otherwise anything inside that one. And also real-time fast mode of ambient ionization. And disadvantages, the matrix in the paper is getting into the mass spec and creating noise, and detection limit is less, and there is a huge background noise which is coming out of the paper. Is it any doubt for anyone until this point? Sorry, is it any doubt for anyone until this point? No, thank you. So I can proceed because like what I'm next getting into is like real what we do. So actually, to looking at the disadvantages of this PSMS, the matrix effect and also limit of detection is less. We want to like think differently. That's why our PI, Dr. Nicholas Manicki, and as well as our senior Magnus Reiberg, think about combining electrokinetic stacking into the paper spray. Then they invented FICP, Faraday ion concentration polarization in paper spray mass spec. What do you mean by Faraday ion concentration polarization is simple. We come across, I think everyone knows about this gel electroporosis. We are just separating the positive analytes or like negative analytes by putting into electric field. It's always similar to that one, but in a different ways. We are using the like different concentration gradient method here. So you look at the right side image C. Here you have we are using a two paper substrate A and paper substrate B. You can look here how we are connecting those paper substrate. There is a reservoir called as R1. We are putting into reservoir R1, we are putting a background electrolyte called as BGE. BGE consists of ammonium acetate with some amount of acidic acid. Then we are spotting our analyte on the paper. Then we are connecting this to the electric field. So by this, what helps? Once you connect it to the electric field with the external voltage of 240 or 300, this one is acts as anode and this one is cathode. From cathode to anode, there is a EOF, electro-osmotic flow is flowing. When anode to cathode, there is electrophoretic flow is flowing. So once these flows are like merge at some point, they're gonna create ion depletion zone. By that one, our sensitivity and selectivity all thing gonna increase. And one of the important thing you need to mention, you need to remember here, we are modifying this paper substrate to build a positive charge on it by using bis trimethoxysiloxane prophylamine just to build a positive charge on it. That helps to reverse the EOF generally. So what's the advantage of this? This helps in actually crude separation. We'll discuss in further slides, and less matrix effect that increases sensitivity and detection limits. This is the main project we are doing currently in our lab. There are 3 to 4 projects going on this, on proteins, on drug abuse using plasma, otherwise urine. In every, like we are 3 areas are doing on this project. So first I can explain the mechanism of how FICP works. So this is the before, like once we spot an analyte, there are both acetate and ammonium ions are there in the entire paper substrate. From the anodic end, there is a electrophoretic mobility. From cathodic end, electrophoretic mobility. Once you put the electric field, once ions are starting moving, there is a titration reaction going on between our acetate ions. The acetate ions migrating from the cathode. At the anode, there is a water electrolysis going on. Those H plus ions used by the acetate ions to produce acetic acid. Once acetic acid is forming, that one is creating ion depletion zone. That ion depletion zone is propagates towards the paper substrate, top of the paper substrate, then that one getting into spray into mass spec. This how practically look like. Look at this image. This one is the most important and careful image we usually explain to new ones who are looking at the who are looking at the our FICP. So once you spot there is a like acetate ions come here and start to neutralize to acetic acid by water electrolysis. Then once ion depletion zone forms, the all the analyte stacks here and ion depletion zone propagates in tip of the paper. Once it comes to tip of the paper, it directly spread to the mass spec inlet. That increase our sensitivity. We can go for less limit of detection compared to PSMS, means paper spray ionization in FICP. So this how the chronogram look like in paper spray ionization. PSMS means paper spray ionization. This how the chronogram look like in FICP. Look here, the entire spray of 1 minute is normal. But here, once the band is moved to the tape, there is a the spray is increased and enhanced in sensitivity. So how the mass spectra look like? This how the mass spectra look like. These mass spectra coming from the drugs in the plasma. Here the drugs are in the noise. The drugs may be like cocaine, methamphetamine, otherwise methadone, fentanyl. These are the class of drugs here. They all are in noise. But once you use this FICP method, our drugs are enhanced in the intensity. That's enhanced intensity helps to go for low concentration in the detection limits. So now the first application is fast analysis using FICP. There are used question. What do you mean by fast in our minds, right? I also don't know when I just moved to the US last year, like when I was new. The fast actually this fluorinated, synthetic fluorinated things, the fluorinated things which is present in water, a polyfluorinated things. These are the most pollutants generally present in low concentration. Generally, analyzing this, people use LCMS method, gold standard LCMS. It takes time and also it takes a lot of money to analyze this. That's why our senior come up with a new idea of FICP to analyze this in a low concentration. You can look at the calibration curve, the image A. Look at the concentrations. It's starting from 10 ppt, 50 ppt, up to 250 ppt. This much low concentration, we can analyze, quantify in the tap water. This is a result from tap water. This paper is already published in ANGI, electrokinetic stacking of free fast analysis. And this how the retention look like. I already mentioned, depending upon the electrophoretic mobility, this chance of fluid separation. And this technique helps for desalting. Desalting helps to enhance your detect the sub PPT level PFAS, but using FICP we can analyze and quantify that one. So next, drug analysis urine. This is actually my project. I done with my project. We are writing paper on this one. This how our normal pictorial representation. This how we connecting our cartridges. This is a top cartridge with the top paper main substrate. This is the bottom cartridge with this substrate. Then this one is a BGE well, R is BGE well. This one is a mass spec inlet, just like half centimeter away from mass spec inlet. We just play with that one, the distance between the mass spec inlet and this paper tip, because we are placing this one on the three-dimensional stage. We can move which direction we want. So toxicology analysis of urine sample, we are doing the direct analysis of urine of drug abuse person. Actually without LCMS, and there is no lot of desalting preparation, salting step, less sample preparation step to analyze this in sub PPT concentration, and which helps to increase sensitivity and include separation and compared to PSMS. You can look at the result. This is a published result I'm showing in ANGICAM by my senior. So the LOD limit of detection compared to stacked means the FICP method versus unstacked. We can go to sub PPT levels to this with this respective drug abuse. Even fentanyl go to 7 ppt. Actually, the real the real concentration in urine is about like 100 ppt, the minimum 100 ppt. This that means in this method we can easily detect in real time by doing ambient ionization with FICP. Then again, I'm going to show how the example spectra look like. How the PSMS is too different from FICP in ambient ionization. You look here, this one, the right side spectra is currently the spectra we are looking at is from the control, means there is no matrix, there is no urine. This control means a methanol water solvent with our drugs. We can see the noise coming out of the some of the unwanted materials here. But by using FICP, look here how the spectra look like. There is no noise at that point when you are doing FICP, when you are analyzing the tape. So that helps to decrease the noise and the unwanted matrix coming into the paper. Then now the spectra looking at the bottom is with a urine, with 100% concentrated urine. Look here, the only two things are showing up. This 114 and 227. Anyone have a guess about 114 and 227? Which is a major component in urine? Anyone have an idea about that one? Sorry. Anyone have an idea about that one? What is 114 or 228? What's the major component of urine? Hello? It's okay, fine.
Creatinine.
Yeah, ma'am. That's right. That's a creatinine. Actually, creatinine is really ionized well. Actually, that's the creatinine. The 114 is a creatinine and 227 is a dimer of creatinine. Both are creatinine is going on. That's why our all analytes are in the noise, completely in the noise. We are not able to see at 1 ppm also. But by using FICP, we can see our drug analytes even in the creatinine presence. Means this FICP helps to just come out of ion suppression also in the presence of huge molecules like creatine. So again, I'm going to show some chronometric data which is taken from the direct from the mass spec. You can look into this. Here, this is just based on the MORZ. You can see this one is a creatinine. This one is this all things our analyte. Again, I mentioned electrophoretic mobility. These analytes are eluting based on electrophoretic mobility. Creatinine is low molecular weight, always have more electrophoretic mobility, elute first, then others elute. So I will show you the two different spectra here. Look at, there are two different spectra. Left one, while creatinine is eluting. While creatinine eluting, this is the creatine. Okay, look at our analytes are in completely almost in noise and less abundance. Once creatine is already elute, when you go to further, when creatine after elute, you can see our analytes are really intense here. This is already I mentioned FICP, Faraday ion concentration polarization is else for crude separation of analyte based on electrophoretic mobility and the molecular weight. This is the just detailed study of that one. How the electrophoretic mobility affects on the coelution, otherwise the crude separation. We can just crude separate the analytes in FICP. Is it anyone have any doubt regarding this? I'm happy to answer any questions because I'm getting into like more tougher things in further slides.
Proceed,
Anyone have
Varadaraj.
asthma? So look here, analysis of drug class like fentanyl, opiates, amphetamine in sub-PPT levels, human plasma. Our last project we discussed about urine analysis. This is all about the plasma. The urine actually contain less matrix compared to plasma. So this my project, I initially try to analyze the plasma directly, means once you get a blood, just separate out blood and plasma, then directly spike the drug analytes into the plasma and directly try to stack it, but it's not possible because the plasma is precipitating and we are burning the papers. Then we done with a precipitation step. We are precipitating the protein using acetonitrile or methanol 2:1 ratio. This already optimization is done. We are just doing 2:1 ratio. Then we once you centrifuge, we can take the supernatant. We are trying to stack that supernatant. Everything same. We are putting some amount of 500 microns Bg into the Bge well. Then we are spotting our that supernatant on these two papers, and we are just putting an external electric field. Then we are stacking using that one. So again, there's a less time consumption method compared to LCMS, because we are not doing any desalting step, and a sample preparation, better limit detection compared to PSMS. This how the detection limit look like. I just mentioned 5 drugs. Probably another 2 months, this paper gonna be out. We are doing this one for 11 to 12 drugs. So methamphetamine in PSMS, you can just go for 3.8 ppb, but in FICP, almost everything go to 100 ppt or 50 ppt. Especially fentanyl, acetylfentanyl, we can detect in 50 ppt. The biological concentration of these drugs in plasma is 100 ppt, but we can go less than that. Actually, this analysis only take you maximum of 30 minutes, means 10 minutes, 5 minutes to extract the plasma from the blood, then another 10 minutes to just do the centrifugation step to protein precipitation. Then once you're done with protein precipitation, then again you do just 6 minutes to analyze this in the our cartridge in front of the mass spec. Next one, this is actually really important thing. This one is done by our senior. So he's writing paper again to the Angie camp. So this one is just protein peptide stacking. How we gonna analyze proteins in this one? Actually, again, protein is only analyzed by LCMS method. That is a gold standard, but it is like offline sample prep is more and it's expensive and slow. But we are doing PSMS, simple and fast, and electrokinetic pre-concentration technique. We know peptides are like shortest simple chains and proteins